Okay, on Monday we were last discussing spinal nerves, which are those 31 pairs of nerves that attach to our spinal cord. We learned that they have two forks on each side, a dorsal or posterior root, which goes to the back side of the spinal cord, and within it are those sensory neurons carrying signals in, carrying them to the dorsal or posterior horn. And then on the other side is where we'll see the anterior or ventral horn carrying motor neurons out by way of anterior or ventral roots of the spinal nerve. Okay, now when those spinal nerves exit from the spinal cord and they're coming together, they're then going to fork again as what we call rami. Okay, these rami, the word rami is plural for ramus, are shown over here. We have an anterior ramus and a posterior ramus. The posterior or dorsal ramus is going to supply the somatic motor and sensory fibers to several smaller nerves, in turn innervating muscles and skin on the posterior surface of our head, neck, and trunk. So it makes sense. We call it the posterior ramus, and it goes to the backside of our body and supplies the backside of our body. Whereas the anterior or ventral ramus goes to the front of our body. It serves muscles and structures of the upper and lower limbs, skin of the lateral and anterior surfaces of the trunk. Now in anatomy, I talk about the anterior and posterior rami, these roots and that kind of stuff too, so hopefully if you've taken anatomy, this stuff kind of rings a bell. We aren't going to talk in this class about the forking that occurs with the anterior rami to form what we call nerve plexuses. So we'll not get into that, but in anatomy, if you remember, we talked about like the cervical plexus and brachial plexus, lumbar, sacral, coccygeal plexuses. We're not going to get into those in this class. Really, that's all we're going to talk for now about spinal nerves. In a couple slides, we're going to look at the function of a spinal cord forming what we call a reflex arc, and we'll focus mainly in on today somatic reflexes that use somatic motor neurons. But we'll also talk about, and this will be towards the end of the lecture, autonomic reflex arcs, which use autonomic motor neurons, which in some cases the spinal cord come from the lateral gray horns, but in some cases autonomic reflexes use cranial nerves, which are up in our brain, as we learned about the other day, or come off of the brain, or coming from our lower end of the spinal cord, what we call the sacral region. So those are things that we'll get into a little bit later. We learned previously that the white matter of the spinal cord enables signals to go up and down to and from the brain, what we call ascending and descending, or sensory and motor tracts. That's the white matter. The gray matter of the spinal cord is a site for integration, for information processing, which is important for our reflexes. Okay, specifically, in this case, we're talking spinal cord, for spinal reflexes, which allows the spinal cord to do the processing and decision making. Okay, we've talked about the hand on the hot stove all semester long. We're going to talk about the reflex that is involved with that. That's a spinal reflex. A cranial reflex occurs in which the integration occurs in the brainstem rather than the spinal cord. So the brain is in charge of a cranial reflex, whereas the spinal reflex is controlled by the spinal cord. Of the two, you probably can guess spinal reflexes are quite a bit simpler. Okay, cranial reflexes involve signals coming up to the brain, and the brain using all those various things that we talked about the other day, doing what they do. We'll talk a little bit about cranial reflexes towards the end of the lecture when we start talking about the hypothalamus and how it plays a role, which is stuff that we've kind of started to allude to back in our previous lecture. Regardless, if it's spinal or cranial, we can describe reflexes as quick, meaning that they happen almost instantaneously. They're involuntary, so they are going to occur without our conscious awareness a lot of times and without our intent. So we don't do them with our conscious well-being and on purpose. They're stereotypes. Anybody have a guess as to what stereotyped in this case means? It means that it's going to occur the same way every time, and if it doesn't, something's wrong. Like when we tap on the patellar tendon, we know what's going to happen to our leg. What's going to happen when you do that? You're going to kick your leg out. Okay? It's not like in cartoons or on Three Stooges where they tap on the patellar tendon and then the guy punches the doctor in the face. Okay? We know what's going to happen, and that's why doctors test for it, because if it's not present, something is wrong. Okay? And the response is going to be carried out either by muscles, which could be skeletal if it's a somatic reflex, or it could be smooth muscle or cardiac muscle if it's autonomic, or it could be glands like sweat glands, oil glands, or digestive glands if we're talking autonomic again. Once again, these are the characteristics of reflexes I kind of just talked about. First thing that's listed there is actually something I didn't talk about, but reflexes don't just occur for no reason. They require a stimulus. Okay? Some type of sensory input is what triggers a reflex to occur. Like tapping on that tendon does certain things to the tendon and muscle attached to that tendon that elicits the whole process. They're quick, generally involve few if any interneurons and minimal synaptic delay. They're involuntary, occurring without intent, often without our awareness, and they are difficult to suppress. Reflexes are stereotyped, occurring in essentially the same way every time. Response is very predictable. We can also classify reflexes based on whether they are autonomic or somatic, which we'll talk more about autonomic towards the back of the lecture, but we'll also talk about those throughout the rest of the semester. These are going to cause excitation, or actually could be inhibition, so it could be contraction or relaxation of our smoother cardiac muscles, or secretion or lack of secretion by glands. Okay. A easy example of an autonomic reflex is what happens when our blood pressure drops and we ultimately cause it to go back up. Okay, illustrating negative feedback. But we will talk about the baroreceptor reflex later in the semester when we talk about blood pressure and the cardiovascular system. Somatic reflexes are the ones that we'll talk about here first. Contraction of skeletal muscle, employing a reflex arc. Now, autonomic reflexes also employ a reflex arc, just a little bit different. What a reflex arc is, an actual potential arcing into the spinal cord, or in the case of autonomic, to the brain. So we're talking spinal reflexes here. So it arcs into the spinal cord, in the sensory neuron, and out of the spinal cord in a motor neuron. Okay, and there are 5 components to a reflex arc, and those are shown here with a picture of a simple spinal reflex. And that spinal reflex is in response to a stimulus. In this case, the stimulus is a tack going into the skin, which is going to cause what sensation? Pain. Okay. We have pain-sensitive receptors in our skin. So those are the first part of our reflex arc, just sensory receptors. Okay. Now, they don't necessarily have to be within the skin. We'll see here in a couple of seconds about reflexes where the sensory receptors are like in the muscle or in tendons or kind of around joints. Okay. But this one just happens to be in the skin. Okay. Now, in this case, the sensory receptors are the dendrites of our sensory neuron. For some of our special senses, we have special cells, like taste cells, that are not attached directly to the neuron, but instead they synapse and send a signal to the sensory neuron. So sometimes it creates a kind of extra step. And this is stuff that we might actually get to see on Friday. If not, we'll definitely see them next week after we've gotten partway through that next lecture. Regardless, the sensory neurons carry the signal into the backside of our spinal nerve, what we call the dorsal root. Here's that dorsal or posterior root ganglion where it's swollen because of the unipolar cell body heads. Coming into the backside over the dorsal or posterior horn of the gray matter, we synapse with the interneuron. The interneuron gets to act as the integrating center in this case, processing that information brought in, deciding what to do. And what do we do in response to a painful stimulus? We take our hand or finger or foot away from the painful stimulus, right? We remove it, which involves contraction of skeletal muscle, which in the case of a somatic reflex is always going to be skeletal muscle. If this was an autonomic reflex, we'd see cardiac muscle, smooth muscle, or a gland. And in order for those to be stimulated, we need a motor neuron carrying a motor signal out. So the motor neuron would be our fourth part. So you can kind of read through all those. Basically just stated them all with our image. So let's look at some examples of somatic spinal reflexes. The first is the one that I've been talking about for a couple class periods now, and that's that knee jerk or patellar reflex. It is a form of the stretch reflex, which is termed the stretch reflex because the initial stimulus is a stretch of muscle. Okay, just that simple tapping of the patellar tendon causes your quad muscles to stretch. It's not a very pronounced stretch, but it is a stretch. Okay. The reflex in this case is called a monosynaptic reflex arc. Mono means one because there's only one synapse that's crossed in the central nervous system, taking place in the spinal cord. Okay. Now, this can occur with the patellar tendon, but it can also occur at the elbow, the wrist, and even the ankle. Okay. But the most obvious and easiest one to test on is the patellar tendon. So we call it the patellar reflex or the knee jerk reflex. Okay. The receptors in this case that are stimulated by the stretch are called muscle spindles. We'll talk more about muscle spindles in our next lecture. These are receptors that surround muscle fibers in our various muscles. And they surround those muscle fibers or muscle cells and detect when that muscle fiber stretches. So the actual signal is a stretch, which creates an action potential in the muscle spindles, which is then going to travel by way of sensory neurons to the spinal cord, which then results in an excitatory synapse in a motor neuron in the anterior horn of the gray matter. If the excitation is strong enough, which means if we hit threshold, action potential will rise in the motor neuron and propagate to the neuromuscular junction, the point where the neuron meets the muscle. Neurotransmitters will be released from the neuron and then will stimulate the muscle to contract. The muscle, in this case being skeletal muscle, will be our effector. In this case, the same muscle that was stretched is the same muscle that's going to contract. So it stretches, so we contract it. It lengthens, so we shorten it. That's the response. Now, in order for this to occur at the same time, we also have to inhibit antagonistic muscles. Okay, like the hamstrings are antagonistic to the quads, your triceps antagonistic to your biceps. Okay, it means they do opposite things. And if your antagonistic muscles are contracted, your agonistic or your prime mover muscles won't move. So if your quads are trying to contract, we can't have the hamstrings contract. Okay, so here's what it looks like. We're first stimulating the whole response by just tapping on that patellar tendon, which causes that quad muscle to stretch. The spindle fibers, or muscle spindles as we call them, are going to surround the muscle fibers, detect that stretch, create an action potential that runs into the backside of our spinal cord. Okay, we're going to do two things. We're going to synapse with a motor neuron that runs back out the front of the spinal cord through the anterior roots, back to that same muscle, which is going to be the effector, causing it to contract. That relieves the stretching. At the same time, we use an inhibitory interneuron to send out an inhibitory signal to the antagonistic muscles, which causes them to not contract. They are then inhibited, so they relax and are leg kicks out. And that's what happens. And think about how amazing it is that this happens as soon as you tap on that tendon. It doesn't take a couple seconds, a couple minutes. It's a direct, quick response. And if you try to hold it back, it's probably not going to happen. You're going to kick out regardless of what you wanted to do. Now, the opposite of the stretch reflex is the tendon reflex. This time, we're going to want to cause the muscle to relax instead of contract. We call it the tendon reflex because the receptors are located within the tendon. Okay, what we call Golgi tendon organs or Golgi tendon bodies, or just tendon organs. These guys monitor tension. Tension that's placed on the tendon. Now, if you're not familiar with what a tendon is, hopefully you all are. Tendons attach our muscles to bones. So when a muscle's contracting really strong, really hard, there's going to be a lot of tension placed on that tendon. If the muscle contracts to the point that too much tension is placed on the tendon, what's going to happen to the tendon? It's going to pop, right? What's something that we don't want to happen, is it? So what occurs in this situation is we get too much tension on the tendon, and so the ultimate goal is to relax the muscle to relieve that tension. So this is a feedback mechanism to prevent damage to muscle tendons. Okay. This reflex is initiated as tension is applied to a tendon, causing the Golgi tendon organs to be stimulated. And they are depolarized the threshold. Action potential is then propagated along a sensory neuron to the spinal cord. Same thing that we just saw. Okay, this time, tension is in the muscle, and therefore the tendon, the Golgi tendon organs are depolarized the threshold, send the signal into the backside of the spinal cord. Okay. This time, our response is to inhibit that same muscle that was contracting. At the same time, we're going to excite the antagonistic muscles. That causes our leg to go backwards, which means that the hamstrings are contracting and the quads are relaxing. And that causes the tension to be relieved in our quad muscle, and therefore the quad tendon. So it's completely opposite of what happens with the stretch reflex. Does that make sense? Our next two reflexes go together, and both of them happen when we have a painful stimulus. And I think they use the example of a tack being stepped on, which we've all stepped on something sharp, and have illustrated these two reflexes. The first is what we do with that same foot or hand or entire limb, and that's withdrawing it from the painful stimulus. We do so by flexing muscles away from the painful stimulus. Here we can show that flexion withdrawal reflex. So we step on a tack. Sensory receptors in our skin are sensitive to pain, create an action potential that travels into our spinal cord. Now you notice that we've got three different segments of the spinal cord shown. We can describe this as an intersegmental reflex arc because we're going to see motor impulses coming from multiple segments of the spinal cord. We actually will bring the signal into the spinal cord. We'll then travel into the white matter where we can carry signal up the spinal cord, down the spinal cord. Remember, ascending and descending tracks are within the white matter. The purpose of that is to activate motor neurons from multiple segments so that the entire muscle is activated. And the muscle that's being activated is our flexor muscle. In this case, our hamstrings. And that allows us to withdraw the limb from that painful stimulus. And you step on something sharp, what you do, you take your leg off of it. Okay. Now, when you do that, you also have to shift your body weight. Okay. Otherwise, you're going to go toppling over, right? So you step on something sharp, what do? You withdraw one leg, catch your body weight on the other leg. In order to do that, this leg better be straight. If it's not, you're going to go down buckling. That's going to cause us to do the cross extensor reflex. At the same time, on the opposite limb, we are going to cause contraction of the muscles that extend the joint to absorb our body weight. So here in this figure, we can combine the two. On the left is our flexor withdrawal. Looks like our right leg. Okay. We are going to withdraw it like we just said. Signal comes in after the stepped on the tack. Now remember, we can cross from one side of the spinal cord to the other side by way of that crossbar. We talked about that the other day. That enables this side of the spinal cord to send a signal out to the other side, to our left leg. This also is intersegmental. Okay, we can see multiple segments sending out excitatory signals to the quad muscles on this leg, causing that leg to straighten out. Our left leg extends. We catch ourselves so we don't topple over. Pretty amazing, all the things that go into just moving your leg, stepping onto your other leg when we step on a tack. All happens instantaneously, and we know all about action potentials and how crazy those are. Release of synapses or release of neurotransmitters out of synapse. So hopefully you really kind of are appreciating how amazing the nervous system is. As far as the exam goes, I will have questions related to these reflexes. You won't need to worry about the images, I promise you that. Just know what the reflexes are, the information about them. That is. So. Now, what I've listed here are some of the common somatic reflexes. We talked already about the knee jerk or patellar reflex, extension of the lower leg. So kicking out of your lower leg in response to tapping the patellar tendon. Tendon muscles are stretched, stimulating muscle spindles, initiating conduction of the two neurons. That's our ankle jerk is just like the knee jerk, except this is with the Achilles, so that's why it's called the Achilles reflex. It is extension of the foot in response to tapping of the Achilles tendon. Tendon reflex and deep reflex mediated by two neuron spinal arcs. Babinski reflex, you might hear it listed as Babinski sign. If you've taken human growth or taken human growth, there's a good chance you'll learn about this. This is in babies under a year and a half. If you basically stroke the bottom of their foot, their toes are going to go like this. Okay, it's pretty fun to do. It doesn't really require anything from the baby other than just the bare foot. But if you show signs of this after a year and a half, it can show that the corticospinal fibers or tracts, which carry signals up into the brain, are starting to be destroyed. So that's not a good thing. Plantar reflex, plantar flexion of all toes, so the toes will curl. Slight turning in and flexion of the anterior part of the foot, response to stimulating the outer sole. Corneal reflex, winking in response to touching the cornea. This is mediated by reflex arcs with sensory fibers in our ophthalmic branch of our fifth cranial nerve. So this is not a spinal reflex. This will be a cranial reflex since it involves the cranial nerves, not the spinal nerves. We involve centers in the pons, motor fibers in the seventh cranial nerve. So if you wink because it's trying to touch your eye, we all do that all the time. That's very, very great illustration of the involuntary nature. If you ever try to keep your eyes open without blinking, do the staring game, it's very difficult. You blink hundreds of times in a day. Abdominal reflex, drawing in the abdominal wall in response to stroking side of the abdomen. It's a superficial reflex mediated by arcs with sensory and motor fibers in T9 through T12. Centers in these segments of spinal cord. Decreased or absent reflex may involve lesions of the pyramidal tract upper motor neurons. So just some of these that you might see. Not super concerned with them, but I'm going to throw them in here. All right, what we're going to do now is transition from talking about the spinal cord and spinal reflexes and reflexes as a whole to discussing mainly the autonomic nervous system. I want to just quickly compare autonomic and somatic. We've talked about this since the first lecture. We've talked quite a bit about the somatic already. Remember, the somatic nervous system is defined as the motor system that controls skeletal muscle. What type of control does it provide us with? Voluntary or involuntary? Voluntary. Good. Whereas the word autonomic, I always say to remember the word automatic because of its involuntary action, providing us with control without our conscious intent or awareness. And this is the division that provides us with control over glands, cardiac muscle, and smooth muscle. We also learned previously the autonomic nervous system is composed of sympathetic and parasympathetic motor neurons and the brain regions that regulate their activities. That's going to be what we're going to talk about for the next several slides. Sympathetic versus parasympathetic. The two divisions of the autonomic nervous system have a property known as dual innervation, meaning they have contrasting effects on the same organ. Okay, what I mean by that is, for the heart, for example, what's the sympathetic nervous system going to do to it? What's the sympathetic nervous system do to us? What's that response that we fight or flight? So what's sympathetic going to do to the heart? Speed it up, make it pump harder. Okay, whereas the parasympathetic producing that rest and digest, what's it going to do to the heart? It's going to go down. Okay, it's going to make our heart rate resting, slow it down. Beat is not going to be very strong, just enough to get us by when we're sitting there at rest. So we have dual innervation, meaning that both divisions innervate the same structure. So how do those structures know when to be sped up, like in the case of the heart, or slowed down? It relates to the neurotransmitters that are released, and that's kind of what we'll get into. Here, probably the last thing that we'll talk about today are the neurotransmitters. We're mainly talking output, because that's what sympathetic and parasympathetic are. But as we know, we always have output in response to input. So the autonomic nervous system does have input that results in the output response. The input for the autonomic nervous system comes by way of autonomic sensory neurons, which we are associating as or with interoceptors. The other name for interoceptors are called visceroceptors. What do you think intero is referring to? Not intero, in this case, think interior or internal. It's a good guess, though. Okay. Viscero for visceral. These are receptors located in our interior or viscera. Viscera is a fancy way to describe organs, which is, those parts of our body are our internal environment. We're going to talk about intero or visceroceptors in greater detail. We might get to it on Friday. We might not get to it till next week. Those are discussed in our next lecture, where we talk about the receptors that monitor blood pressure and blood pH. We actually have some in our brain that monitor our blood glucose and our water balance. So we'll talk about those receptors called chemoreceptors in our next lecture. But for now, let's talk output. And that's the sympathetic, parasympathetic stuff we've already started to talk about. The activity of the autonomic motor nervous system is going to regulate visceral activities either by increasing or decreasing their activity. Or we can say exciting versus inhibiting. their activity. And once again, for autonomic, the effectors are always going to be cardiac and smooth muscle and glands. As opposed to somatic motor neurons, which are usually myelinated and travel from the central nervous system all the way to skeletal muscle fibers, autonomic motor pathways consist of two motor neurons in series. So I'm going to jump ahead a couple slides to show you a picture of what I'm talking about. Here's what, at the top, the somatic nervous system looks like. Okay, we've got one motor neuron that runs from the front of our spinal cord out of the anterior root of our spinal nerve all the way, and this is a myelinated neuron, to our effector, which we know somatic, the effector is always going to be skeletal muscle. Look at this second image, and we're going to skip this one for now, and the third image. For both sympathetic and parasympathetic, running out of the spinal cord, we've got a what we call preganglionic neuron that synapses with a postganglionic neuron. So instead of going all the way from central nervous system to the effector, we go central nervous system to what we call autonomic ganglion, and then to the effector. So we've got a pre and then a post. What does pre stand for? Before. What does post stand for? After. So the first motor neuron before the ganglion is called the preganglionic neuron. Guess why we call it preganglionic. The second motor neuron is the postganglionic because it's after the ganglion. Okay. We say it's a point of contact, but we know neurons never technically contact one another because the dendrites and the axon terminals never actually touch. There's that small gap called the synaptic cleft that exists where we release neurotransmitters, and that will activate or inactivate the next neuron. Okay, we're going to be looking at activating in this case. Okay. So the preganglionic neurons can be described as extending from the central nervous system to the ganglion. Then from there, the postganglionic neurons extend from the ganglion into the peripheral nervous system to the effectors. So let's look at sympathetic and compare it to parasympathetic. We know that sympathetic nervous system or division causes that fight or flight response. So the other name for sympathetic could be fight or flight division. This is what adapts our body for physical activity, which physical activity, we're running or fighting. Those are both forms of physical activity. Doing things like increasing heart rates. We're pumping blood faster and more of it. Our blood pressure is going to go up. Our airflow into our lungs will go up. We'll increase our blood glucose levels to make more ATP. We'll increase the flow of blood to our cardiac and skeletal muscles. Doing everything that adapts us to, run away from a bear or stay there and fight it. Okay. Now, it doesn't have to be as, crazy as a situation as, a bear attacking. It could be just as simple as, trying to exercise. We're going to have to kick in this fight or flight response to exercise. Okay. Now, output for the sympathetic nervous system comes from this region of our spinal cord that I was talking about the other day. Okay, T1 to L2, from the top of our chest down to the bottom of our chest, basically. Okay. And for that reason, we can also call the sympathetic division the thoracolumbar division because it involves the thoracic and lumbar regions of the spinal cord. Remember how I said that those lateral horns were only found in that part of our spinal cord? Go back to that picture. Okay, this happens to probably be a thoracic region or top of the lumbar region. Here's where we can see an autonomic motor neuron. This would be a preganglionic neuron of the sympathetic nervous system. So that's the first neuron that comes out of the spinal cord. And with the sympathetic, that first neuron is going to be really short. It runs to a chain of ganglia or trunk of ganglia on both sides of our spinal column. We then synapse with the second neurons, those postganglionic neurons or fibers, which are going to be much longer because they have to run the rest of the way to the target organs. Let me show you a picture of what I'm talking about. I know this is a couple slides ahead, but I wanted to show you. Okay. Here's the spinal cord. Here's T1 through L2 or L3 it shows. You can see there are, and we're just showing on one side, neurons leaving that spinal cord. What we're not showing are the vertebrae that surround it, because that would just be in the way and we wouldn't be able to see anything. Right next to those vertebrae are these chains or trunks of ganglia where we're going to synapse between the first neuron and the second neuron. See how these little dots in here? Those are the cell bodies of the postganglionic neurons. So the first neuron is going to be really short, the second is going to be really long because it runs the rest of the way to the target organ. The parasympathetic division utilizes the brain and the spinal cord. It utilizes cranial nerves, not all of the 12 pairs, and it only uses the bottom little bit of the spinal cord, what we call the sacral region. So the other name for the parasympathetic nervous system is the craniosacral division. Cranial for brain, cranial nerves, 3, 7, 9, and 10, 10 being the most important. At this time, the pre's are really long and the posts are really short. Here we can see an image, the same image I've already referred to a time or two, to illustrate this difference between sympathetic and parasympathetic. We're going to wait and not talk about this one quite yet, but the preganglionic neuron is really short. For the sympathetic, post is really long. Here for the parasympathetic, it's really long, but then it's really short. Something else that's very different about the parasympathetic is it doesn't use those chains on both sides of the spinal cord. It does something different. Now this third image shows that the sympathetic nervous system also incorporates the release of some hormones from the adrenal medulla. And I'll get to that here in a second. First, I want to just describe what the concept autonomic tone means. It describes the normal rate of activity representing balance between sympathetic and parasympathetic. Think of these two things like a teeter-totter. As one side goes up, the other side goes down. Right now, I would say that the top of your teeter-totter is parasympathetic because you guys are all resting and digesting. If I told you guys put your notes away, we're going to take the test in the last 10 minutes class, what are you going to do? You're going to flip it. Okay, sympathetic's going to get active, your heart rate's going to go up, you're just going to sweat, you're going to breathe real fast. Some of you might want to flight, run out of the room. Some of you might want to charge at me and fight me. Okay, just shows you how nice and amazing that is. We can turn it on a dime. Okay, on a second, millisecond type situation. Fortunately for you guys, I'm not going to do that to you. I've already alluded to some of the information on this page with the figure that's on the following page, where I talked about that sympathetic trunk or chain of ganglia where the synapsing occurs between the pre and the post. Not always does that occur, but most of the time this does happen. The figure here on the next page shows that. Okay, if I just show you T1 here, runs out. So this preganglionic neuron synapses with several different postganglionic neurons. This one happens to run to the trachea. Okay. What's cool is sometimes, like we showed down here lower, like T8, for example, runs into the chain, and then it can fork to go down or up. These forks and axons are called axon collaterals. Okay, and so this allows synapses to occur at different levels of the chain of ganglia. Okay, in our abdomen, some preganglionic axons will actually pass through the chain without synapsing. Instead, most of these synapses with postganglionic neurons in what we call collateral ganglia, which are located closer to the digestive tract. Here we can show what I'm talking about. Let's just show this celiac ganglion. So for T6 and several of those right there, they travel directly through. There's no cell body here. So they travel through that ganglion to another type of ganglion where the synapsing does finally occur. The same thing exists though. You have a pre and then a post that then runs to the effector. It's just the synapses outside the chain. But for everything kind of up by our chest, it's going to synapse in that chain, okay, without passing through first. Okay. There is another exception, or there is one exception to this rule, which involves the adrenal medulla. I'm going to go back to that figure here on that previous page, showing you this third part that I told you I was going to talk about in a second. No matter what, whenever we activate that fight or flight response, we're going to send signals by way of preganglionic neurons to the adrenal medulla, which is the middle part of our adrenal gland, which is located on top of our kidneys and our abdomen. Inside of our adrenal medulla are chromaffin cells. These are cells that release epi, norepi, and to a lesser extent, dopamine. The main one being epi, which I said the other day is also known as adrenaline, which is where that adrenaline rush that we talk about comes from. The great thing about the release of epi into the blood is it's going to distribute that epinephrine all over the place. And widespread delivery of epinephrine is going to help produce that fight or flight response. Same thing for norepi and dopamine. Okay, so the last bullet here is something I just said. Release of epinephrine as well as norepi and dopamine to a smaller extent accompany activation of the sympathetic nervous system. This does not exist for the parasympathetic. The parasympathetic nervous system does not have a chain or trunk of ganglia either. Instead, what we see is that preganglionic neuron running almost all the way, or in some cases running all the way to the target organ, and then synapsing within what we call terminal ganglia, right there within the organ or right next to it. So the pre is really long and then the post is really short. And we can see that here on the next page. You can also see that we don't really involve much of the spinal cord. Just the very end in the sacral region. A lot of the signaling actually comes from some of our cranial nerves. There are 4 pairs, 3, 7, 9, and 10, with 10 being the big dog, the vagus nerves, because they carry out 80% of our parasympathetic nervous system signaling. Around 80%. You guys remember what Vegas meant? Not as in Las Vegas, but in this spelling. It meant wandering, because these are so long. They travel from our head, through our neck, through the thoracic or chest cavity, and then through the diaphragm to the abdominal cavity, which makes them probably several feet in length. Look how far down they can go to our intestines, which are, pretty far down in our abdomen. Okay, so those preganglionic neurons can be super long, and you can even just barely see those short postganglionic neurons. Okay. The cell bodies of preganglionic neurons and vagus nerves are located in the medulla. Their axons extend into the organs of the thoracic and abdominal cavities. 4 pairs of cranial parasympathetic nervous system ganglia innervate structures in the head. Other postganglionic axons of the parasympathetic nervous system innervate heart, airways, lungs, liver, gallbladder, stomach, pancreas, small intestine, part of the large intestine. So it makes sense, hopefully, that for the head, all the innervation for the parasympathetic nervous system is going to come by way of some of our cranial nerves. It wouldn't make sense for sacral stuff to come up through our head or through our chest into our head and innervate our face. Okay, that's going to come by way of neurons or nerves in our head. Okay. Now, this does kind of defy, standard logic with the vagus nerve running downwards, but really, being kind of straightforward, is what the sacral region of the nervous system does, or the spinal cord does. Innervates those lower organs: colon, uterus, or ureter, urinary bladder, reproductive organs, which would include the uterus in females. Now, you might be wondering, how does the heart know to speed up when the sympathetic nervous system is active, and how does it know to relax when the parasympathetic nervous system is active? It relates to the neurotransmitters that they release. Okay. Different neurotransmitters are secreted at the target organ, and the target organ is going to have specific receptors for those various neurotransmitters. What was our most prominent neurotransmitter? Acetylcholine. I've also heard it pronounced acetylcholine. I had an instructor that talked about it that way, or pronounced it that way. I've always said acetylcholine. ACH. This is secreted by most of our neurons. Okay, if I show you a figure here, it's always secreted by preganglionic neurons. Doesn't matter if it's sympathetic or parasympathetic. The first neuron, always acetylcholine. For the sympathetic, it also secretes it by the postganglionic neuron if it's innervating a sweat gland. For the parasympathetic, that postganglionic neuron is also going to secrete acetylcholine. So parasympathetic, it's acetylcholine, acetylcholine. For sympathetic, when we're innervating sweat glands, it's acetylcholine, acetylcholine. Here's the difference between sympathetic and parasympathetic. The postganglionic neuron, for effector cells, that would be like other glands, smooth muscle, skeletal or cardiac muscle, norepinephrine is what's secreted. Okay. The receptors involved in receiving acetylcholine are called cholinergic receptors, and it includes two different types, what we call muscarinic. We find those on cardiac muscle, smooth muscle, and glands. I always think muscarinic because it's on the muscle. Nicotinic, those are the ones on the postganglionic neurons, specifically their dendrites, as well as the chromatin cells in the adrenal glands and at the neuromuscular junction, which illustrates the fact that, and if I go back several slides here, we see acetylcholine being released by the somatic nervous system. So if I ask you on the test, which hormones being or which neurotransmitter is being secreted by this neuron, good chance it's acetylcholine, 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 acetylcholine. There's only one instance when it's not, and that's when we release norepinephrine. Go ahead and write this stuff out, and then we'll just pick up with this first thing on Friday.